and Dr. Pradeep Tarabdar. He mentioned that he is my collaborator and friend, but I consider more of my friend than the collaborator. We spend a lot of times on the tea and walking around the campus, and we work together after we came to know each other. So, and this is a very good initiative. I remember in 2017 or 18, I was actively engaged with iGEM, and eventually in 2020, I believe, they come up with a proposal for discover for synthesizing some sort of strategies of proposing some strategies regarding mastitis in bovine. Based on my expertise and my fascination to work with animals and human interface, I'm very much inclined to help to offer the supports or decipher the knowledge whatever I have with respect to benefit of animal health. So again, this time I was abroad, so I could not make my time to get actively engaged with this IGN, but I offered my all help and support, and I'll continue it. So with this, let's see what I can give
So,
you today.
Professor, can you hide it? Stop sharing hide button.
Okay, great.
Yeah.
I can do that. And also let me take my pointer. Yeah. So today's title, as you know, that bacterial EVs or extracellular vesicles, and I will take you to the path where we can see the what is multifunctional, what I'm trying to mean, and how it can be utilized as a biomedical models application model. So I think Pradeep already mentioned my journey from my undergrad, where I did my veterinary, and after that I did my masters in microbiology, and then I did my PhD in biotechnology, where main focus was molecular immunology, and then I did my two postdocs, one in University of Guelph, where I tried to explore some sort of virus-like particles-based vaccines against avian influenza, and then I moved to University of Toronto for another postdoc, and then I joined ISR Kolkata in 2014 with an ambition to start and understand the host pathogen interaction better, in the sense of, there are many pathogens around us. We cannot understand all of their involvement in host and pathogen interactions. So but broadly, if you can see that these are the right panel where you can see that different modes, different way that host and pathogen interact each other, and what are the different variables or factors which affect this interaction. So my focus is actually understanding a few of them, because this is a very broad area of research. For example, one of the major focus for my research group is how actually host susceptibility make difference against a particular pathogens. Also, I focus on how route of entry of pathogen makes difference. So all of them means host pathogen interactions, which is what is the outcome. The outcome is either immunity or infections. So immunity means you are showing the sort of responses which protect our body and protect our cells from further infections or further damage. On the other hand, if our immunity fails, then we all know that infection starts. But as I said, these are the different variables, and these are the key factors which determine this outcome, whether it will be a immunity or it will be infection. So there is a fine balance which we are trying to understand, taking into very few models. So if you see the host pathogen interaction, how it evolves, and there are so many reasons which are actively, directly or indirectly influence the outcome of these interactions. For example, our behavioral changes, human habitat, encroachment of the wildlife, and the other, from urbanization, better urbanization, industrializations, all those affect these host pathogen interactions, which you know that, for example, all those pandemics, they're basically changes with human behavior and social and economical, uh, socio-economical stature of the current practices. Other than that, you see that there are a lot of industry, a lot of problems which happens due to the antimicrobial resistance. We all talk about it, and but the main problem we never identified and we never resolve it. The problem still there. So one of the problems that arises from the AMR, for example, for us as a human, is basically injudicious use of antibiotic in animal husbandry practices, which from my personal attachment to veterinary science, I know for fact for antibiotic use is not only for disease prevention or disease control, sorry, disease control. It is also being used for different purposes. For example, some of the antibiotics helps if you use suboptimal dose, they can activate the growth, they can make more bodily shape like meat or milk production increase, which is of course not good. And the meat and the milk also contain the residual antibiotics, which ultimately affect the environment. So if you talk about the different types of the disease, out of 1400 different species, more than half of them are called zoonotic disease. These zoonosis, you know that the disease which freely transmit from animal to human or human to animal directly or by indirectly using different vectors. And out of them, there are few disease which we call emerging infectious disease, which is one of my interesting area of research. So at one point, let's say 1980s, we thought that many of the disease which are currently still we are having the problem or issue with them, they are they are eradicated. But with the effect, with the change in the human behavior, with the change in our food habits, with the change in industrialization, urbanizations, those diseases again emerge. So we call them emerging infectious disease. One of them are all those viral pathogens which we see often as a pandemic form, like SARS-CoV-2 or even influenza, H1N1, H5N1. They're all kind of emerging infectious disease. And most among in EIDs, there are many of those EIDs are considered as a gut. They causes the gut disease, gut associated disease, or gut and respiratory system associated disease, which uses mucosal route as a primary portal. What is mucosal route? As we know that mucosal route means we are talk about nasal route, oral route, conjunctival route. They're all kind of considered as a mucosal route, and the pathogens which enter and invade our body are normally known as mucosal pathogens. So with this brief background, the primary focus of my research is basically understanding the molecular mechanism of host pathogen interactions, and of course how to handle these interactions. As I said, whether it's infection or immunity, if it is immunity is good, but as infection we need to be very careful about it. So we have been trying to develop some effective measures to control them. Well, for my convenience, I picked up two specific models, viral models and bacterial models. I'll come to you later. So we try to understand the host perspectives, because infection and immunity is always we cannot blame the pathogen. It also the host status, right? Their susceptibility, route of entry, environmental factors, their food habits, how the gut health is, all matters from the host perspective. And from the pathogen perspective, it's very well known that different virulency factors and with the time the microbial evolutions, which you know that there are different subtypes and which are evolved and making mutants, and they poses bigger risk than what we thought. So one of the approach, so now problem we identified, so how we are trying to address it. So we are trying to use some sort of very conventional mode where we used bacteria as a vector or bacteria as a delivery strategy, live bacteria. So where we choose to use normal probiotic bacteria that we commonly use for milk and milk fermentations, which is lactic acid producing bacteria. We focus on bioengineering of those vectors, able to express heterologous proteins, because they have their own set of proteins, but they are kind of the lactic acid producing bacteria are normally offer us a non-specific immunomodulations or the non-specific effect, which is a general effect. But if you want to have a targeted effect plus generalized effect, then probably the benefit would be much more, we can see larger scale, because if you use them as a mucosal mode, then in addition to their natural adjuvanicity or natural beneficial effect, we can also achieve a targeted benefit. For that, we use different strategy how to bioengineer those bacteria. We use, let's say, depending upon the purpose of our use, those bacteria are so versatile that it can be used as a secretion system, or protein that need to be secreted into the extracellular milieu, or if you want the protein to be expressed or retained on the cell surface of those bacteria, we can do it. Means already those technologies are available for the other bacteria, but for the probiotics, I think it's very interesting considering their normal natural benefits. And the other way we are trying to, in other part of my research, where we focus to understand the putative virulence reporters of the gut pathogens, and how understanding and studying those virulence factors, how can we improve the gut health, because normally they're supposed to infect our gut and affect the epithelial cell, disrupt the tight junction, creates gastroenteritis, diarrhea, and other illness. So, because studying the gut and with the pathogen is very complex niche, because it's the residence or it's the habitat for lots of bacteria, millions of bacteria. They stay together. So there is always a interbacterial competitions, which is for a large scale it is possible, but we started from the very scratch to understand the host pathogen interaction inside the gut using very few bacterial component and very few bacterial types, where we particularly focus on bacterial toxin export system, how it affects the host, whether the toxin export system of the bacteria have any other advantage. And then it comes another way that many of the bacteria, almost all the bacteria, as a canonical end product, and is a highly conserved, highly conserve things, that they produces some sort of vesicles. That is the main part of today's talk. So this is very ubiquitous in nature. Almost all the bacteria and all the mammalian cells also produces some sort of vesicular structure at different stages of their growth or cell cycles. And also some bacteria, particularly which are beneficial bacteria, we consider them. For example, all those lactic acid producing bacteria, lactobacillus, lactococci, they produces some bacteriocins. They are small antimicrobial peptides, and they help maintain our gut homeostasis by killing or targeting the virulent or pathogenic bacteria. Okay, so let's take you to what we know about extracellular vesicles. You know that extracellular vesicles are, as I said, it's ubiquitous in nature. It's all over. All the living creatures, living cells, they produces EVs at particular stage of their life cycles or growth phase. For example, archaea. So this is the archaea, which all those black dots are representative of EVs, extracellular vesicles. Gram-negative bacteria, gram-positive bacteria. You talk about mammalian cells, which normally we call them exosomes, and also plant cells. If you grow them, the culture supernatant, you can always find a lots of extracellular vesicles, which resembles the architecture, or resembles very compositionally with the source bacteria or the source cells. So the discovery of the vesicles, this kind of vesicles, is not new, and you will be proud to know that the first time the extracellular vesicles was demonstrated by S.N. Chatterjee and J. Das uh, from Tropical Medicine in Kolkata. So in 1967, while they're working with Vibrio cholerae, at that time they were unable to understand what are those vesicles, but electron microscopy captured those vesicles. And then some of the further works, which is in 1990s, they also electron microscopically they demonstrated that they also contain some sort of nucleic acids. But all are confined to the gram-negative bacteria. Nobody has tried in that extent with the gram-positive bacteria. But eventually, the research progressed, and as I said, the outer membrane vesicle production from gram-negative bacteria or from the mammalian cells is not new anymore. But there are many aspects or facets of those OMVs are still need to be studied in depth, because These are the potential biological functions that they can exhibit when you can use in different way. For example, they can carry the nucleic acids. They can, where is the nucleic acids? Yeah, they can carry some nucleic acids due to horizontal transfer, and they can have different enzymes or metabolites which cells doesn't like or cell wants to vomit out. So those can be present, those can be encapsulated or carried by those outer membrane vesicles or extracellular vesicles. They can have different virulency factors which normally the source bacteria from where they originate, they can carry. They also help for the intra or intercellular communications, which is very interesting. And then there are recent research also support that OMV or EVs or bacterial vesicles actively involved in biofilm formations, so they can project themselves as a protective barrier for the bacteria. So the disease which we always know talk about, hard to treat infectious disease, so obviously those EVs contribute to that phenotypic attributes or pathogenic attributes of the bacteria. Now, how they can be used, there are lots of literature and lots of work around the globe, so already published, and and the researchers around the globe are still trying to fine tune their applications. So starting from the vaccines, so why vaccines? As I described, that they can represent the bacteria as a canonical end product, non-proliferative. So that is one of the advantage for the EVs. So you can use it just as a vesicule which contain your proteins and activate the immune systems, because the cell surface is very similar. I'll discuss in the later slides how the OMB biogenesis happens. So we can clearly, we can clearly, see that their surface will be decorated with the similar type of proteins which are normally a bacteria represents on their surface. So these and the surface antigen are mostly immunogenic, and because of their accessibility and virulency mechanisms, they can also be served as a good adjuvant, because they are membrane lipoproteins, for then peptidoglycan, different components of the bacteria which are non-specifically recognized by our innate cells, and also flagella, lipopolysaccharide, there are TLR ligands. So if they carry them, they can also serve as a adjuvant in addition to the vaccine candidates. Their cancer immunotherapy is a budding field where people around the globe are working on how these OMVs collected or from T cells or B cells or immune cells can help for cancer immunotherapy. And this drug discovery is just like a normal nanoparticles, where most of the, I think Pradeep also very much in this field, that vesicle structure can easy to encapsulate any drugs based on the water solubility or on non-solubility. We can entrap those drugs into either the lipid bilayer or in the inner core of the vesicles, which is water soluble content for that. And then recently some work that proposed that those can provide a additional barrier for the epithelial cells, because they have the receptors, because they have the ligands which can bind to the cell surface of the epithelial cells, and they can coat that. It's just like a mucus. So if they coat that, the source bacteria, they cannot bind to the epithelial cells, the target receptors or target cells. So they possibly can help to prevent the gut pathogen from entering into the epithelial cells or their target cells. Now, I'm sure means this is a very nice slides, and you all know about it. Just I want to describe a little bit so that we understand what are the compositional aspect and how the biogenesis depends on the cell wall structure outer of both gram-positive and gram-negative bacteria. For example, gram-positive cell wall, you all know that they're thicker than the gram-negative, and because of the peptidoglycans, these peptidoglycans are decorated on the outside of the periplasmic space, and they have different lipoteichoic acid and teichoic acid all together give a very strong conformations of the cell wall, which make them more robust cell wall structure in case of gut pathogens, in case of the gut residents. While in case of the gram-negative bacteria, there are outside of the gram-negative bacterial cell wall, there is outer membrane which are decorated or which have abundance of lipopolysaccharide or lipooligosaccharides, and they have plasma membrane, bilayer plasma membrane, very similar to gram-positive. The only difference is, that in the periplasmic space, they have very fine layer of peptidoglycan, while in this case, the gram-positive, they have a thick layer of peptidoglycan. And that peptidoglycan bilayer, that peptidoglycan layer, determine the OMV biogenesis and the mechanism of OMV biogenesis, how the OMV or EVs comes out or bud out from both type of cells. And based on this compositional difference, their outer membrane vesicles or EVs compositions also change because of peptidoglycan. So they have some at some point they can have the peptidoglycan as well, but they only composed of the plasma membrane, which is this inner plasma membrane, not the outer one, because they don't have that. But in case of the gram negative, the EVs composed of the outer membranes, so they are more, in terms of thickness, they are more thinner than the gram positive vesicles. But other aspect which we can clearly differentiate from the virulency aspect, since gram positive bacteria, if it is a, let's say, if it is non-pathogenic, then there nothing to carry a pathogenic component. But the gram negative cell wall, since it contains the lipopolysaccharide, the EVs which are secreted by the gram negative bacteria are little bit risky in terms of carriage of lipopolysaccharide or lipooligosaccharide. They can be good adjuvant, but it in higher content, higher amount, they're highly endotoxic. But the same thing when applies to the gram positive, they are safe, but it's a difficult process. As you can see, to disrupt this peptidoglycan bilayer is not a easy job for them. Now, if I come to the how the OMP biogenesis happen in case of the gram-negative bacteria, there are mainly three steps. One is unbudded gram-negative bacteria. It started to bud out from the outer membrane, and then outward bulging, and finally it pinched off from the membrane and comes to the extracellular milieu. So there are several mechanisms being proposed, but they are all in a stage of hypothesis. So one of the hypotheses or model we can see here is the reduced envelope crosslinks. As I said, in case of the gram-negative bacteria, they are very thin peptidoglycan bilayer. And this peptidoglycan is cross-linked with LPP lipoproteins and different peptidoglycan protease. If they do not function well, these cross-links disrupted or get loosened. So that creates a kind of formations of bulge, and that can be bulged out from the bacteria. And the other mechanism is the accumulations of different metabolites, different peptidoglycan fragments, proteins, aberrant proteins or misfolded proteins. They can accumulate and increase the turgor pressures, which leads to pinch of the vesicles along with the outer membranes. The third model, where there are different lipids which are extracellularly comes or which are present in the outside of the bacteria where they grow, there are different lipopolysaccharide associated proteins and the bacterial and the branched fatty acids. So if they're available in the extracellular milieu of the bacteria, in the gut particularly, they can also increase the bulge formations and turgor pressure from the outside, which also helps to take it off or pinch off from the bacteria. In case of Pseudomonas, they have special, special way or the specific component which is Pseudomonas guanylin signals, which comes from the outside, which also helps for bulging out. But this has been observed only in case of the Pseudomonas bacteria. And there is another mechanisms which has been proposed is downregulation of BACJ, YRB, and ABC transporter. All three together helps for the protein import inward directions from the outer membrane to the inner membrane. But if somehow, due to some stress, metabolic stress, or other unknown factors, if it downregulated, then lipid transportation is not possible. Inward lipid transportation is not possible, which over the time increases the target pressure and the OMV formed. So as you can see, the difference with the gram-positive bacteria, the main difference is actually lies here. In peptidoglycan, it has a outer wall zone and inner wall zone. So outer wall zone and inner wall zone is basically the abundance of the peptidoglycan links. So this is little bit tough, and this is little bit lighter in terms of thickness. So there are three, normally what happen again, due to some metabolic stress, unknown factors, the target pressure increases, or in the cell cycles. These increment happen only in the positions where some of the specific phospholipid enrichment happens, which leads to curvature bind, which leads to make a curves, and these curvature binding proteins are also responsible for that. And within those vesicles, within those bulged formed, there are many proteins, as I said, the metabolites, nucleic acids, fragmented nucleic acid, DNA, RNA, they can accumulate there, which increases the pressure, and it can bulge out and stay in between this periplasmic space. And over the time, autolysing damage those peptidoglycan layer, or loosening the integrity of the peptidoglycan bilayer. So finally, they comes out of the cells and released in the extracellular space. So if we combine these things, there are two major mechanisms, where the membrane bleeding, and then there is EV precursors in the inward in inner wall zones, where it can release by two methods. One, either it can burst the peptidoglycan layer and comes out with the outer membranes, inner membranes. And the other mechanism is it can come along with the peptidoglycan, so the vesicles can be coated with the peptidoglycan. The second mechanism is that inward, so the inner wall zone of peptidoglycan, it expands and it explodes. When they explode, all the cellular contents along with inner membrane, they comes out as a vesicles, and this is and then they serve as a EVs for the gram-negative bacteria, where we hardly see the peptidoglycan layer coating or coating the vesicles. But the problem of studying these bacterial EVs, there are lack of optimized protocols. We can always get EVs from the bacteria, but you imagine a bacteria growing in 200 ml of culture, and you are trying to get the EVs to be isolated from this culture supernatant, which is not an easy job. Means it needs lots of medias to get the required amount of EVs. So we definitely we realize that we need an optimized protocol to start with this work, and then their heterogenic nature, and they're compositionally diverse. As you can see, the gram positive from the gram negative, even from the same gram positive bacteria or gram negative bacteria, it's different from one bacteria to another bacteria based on their toxin factors and the surface architecture, other appendages like flagella, cilia, or we call it pili, capsules. So all those matters. So we should not expect a similar or ubiquitous type of composition in the EVs, particularly when they're from the different origins of bacteria. So to study that, we took both type of bacteria. One is Campylobacter jejuni, another is Lactococcus lactis, because we already have established this Lactococcus lactis for our purpose. As you know, my purpose is to bioengineer that. So we took this one as a model, as a gram-positive model. And Campylobacter jejuni, they have spiral sepate bacteria, very nice. Nice shaped bacteria, which only thing you cannot see the flagella, which due to, I don't know, we never got the flagella. They are bipolar flagellas. We expect a flagella this side, flagella this side, but for some reason we are unable to get the bacteria or culture the bacteria, image the bacteria with the flagella. But yeah, they are Campylobacteria. And if your cell membrane structure, cell wall structure of the Campylobacteria, in addition to the common gram-negative representation of the common gram-negative bacterial cell wall, they have a kind of flagella and secretion systems. So this is a little bit unique sequencing system, which is not commonly found in all bacteria, all other gram-negative bacteria. Very few bacteria so far was found to harbor this type of sequencing systems. And in case of the Lactococcus lactis, this is the peptidoglycans, and the peptidoglycan in between give the conformity and the strength. They have lipotiquic acids and other components, and polysaccharide pellicles. They all make the peptidoglycan bilayer very stout, and then there is a cell membranes. So the protocol wise, you can see in different papers there are different protocols. The one of the simplest protocol to do the EV isolations is growing the bacteria, but we need to monitor the bacteria at what stage we should collect the bacteria or stop the growth. At what stage the EV production is high. So far, what we understand that at exponential stage, growth log phase, the EV output is better. So after you collect the bacteria, separate the bacteria by filtrations, we go for ultracentrifugations when we get pelleted, and further purifications of the specific OMB pellet, because we do density gradient, and we can do further purifications by using different gradients and multi-gradients tubes, and then different fractions we can collect, and we can check which fractions actually have a good number of EVs. So once we isolated those EVs, filtered, we make sure that EVs do not contain any component of bacteria like debris or fragmented flagella. That's why this step actually helps the gradient centrifugations. And when you micrograph, electron micrograph, you can see the size, which is very clearly vesicular size, and the lipid bound, they are lipid bound vesicles. I'll tell you later, but their size is vesicular, but they are not uniform. They're kind of a different shape, different sizes, but the range of those EVs, particularly we get it from Campylobacteria, is I think 75 nanometer by ACM we imaged, which is 55 nanometer by 10 and 103. So you can always consider them as a nanosite structures, which ranges from 50 to 100 to 200 nanometer. And since they are extracted out or they are released from the gram-negative bacteria, their net surface charge is obviously negative, which is minus 19 millivolt. And when you see for the analyzed by for their compositional study, we did mass spectrometry, LC-MS, and where we identified a huge number of proteins, out of all of them are categorized into 20 different orthologous groups, and we tried to identify what are those proteins particularly responsible for host pathogen interactions, like surface expressed proteins, because those proteins are different subcellular fractions of the bacteria. So approximately out of 65 proteins that we have identified, the majority of the proteins are hits are from periplasmic, membrane associated, cytoplasmic proteins. And also interestingly, as we saw, I showed you before that the Campylobacteria has a sort of types of sequencing systems. We are interested to see whether they can also carry some segments or some subunits, either intact or fragmented, into the fraction of OMVs. And we can see that one of the proteins, which is VGRG proteins, it is one of the effector proteins for type 6 sequencing system, which is unique for Campylobacteria. We can also get the signal, or we can also see the proteins in mass spectrometry. Now, this compositional and the biophysical characterization has led us to study whether, as a nanosized lipid-bound vesicles, are they able to how they interact with the host cells. So for that reason, so we try to, I don't know why it's making sound. So we try to first understand what would be the concentration of OMBs that we should use for intercellular for cellular interactions. So we optimize we use the denser concentration dependent uptake assay. So with this 10 microgram, 5 microgram, there's actually the total protein content. So it's not the other composition, other components like lipids. So it's the concentration is based on the proteins. So we saw the concentration dependent increase when we co-intubate those OMVs or EVs with Campylobacter jejuni, and jejuni with human, I guess this is human INT407 cells, and you can see that the increment in the OMVs, intracellular localization of the EVs. So we then picked up a particular concentrations and then used them for a time scale, a time-dependent uptake process, where we started from 15 minutes and went up to 24 hours. And you can see the maximum uptake start means it reached to the maximum kind of 24 to 20, 12 to 24 hours, which is reflected in the flow cytometric patterns with histograms, and the clear shift in the right of the cells, which indicates that is a concentration dependent uptake and a time dependent uptake of the EVs, which tells that EVs are not just naive or very benign things. They can be uptaken by the host cells and they can co-localize inside the cells, but not sure where, either cytoplasm or in the other organelles. Now, what is the mode of this uptake? So, as a nano-sized vesicles or nanostructure, it is very well anticipated that they will be uptaken by the endocytosis process, which is in fact true. The different pathways we try to understand which pathway the endocytosis happen, what would be the major pathway for intracellular perfusion or intracellular uptake of EVs or OMVs. So by using, so this is a very interesting means we learn basically in our lab. It's a well-established methods to determine the endocytic pathway. So where we try to block the different specific endocytic pathway using clarithrine and caveolin inhibitors, which is dinosaur, and then nystatin, which is lipid wrapped inhibitors, and then cytochelacin D, which inhibit the macropinocytosis, amiloride, which also uh, inhibit the sodium and hydrogen exchange. So all those pathways, when we checked with specific chemical inhibitor like dinosaur, nystatin, and cytochelacin on amiloride, we observed that the dinosaur actually block the most of the EVs to get internalized, while the other pathway, for example, lipid wrapped or macropinocytosis, it's not that much, the blocking is not effective. So we come to a point that dinosaur would be the one of the blocker which is dynamic dependent. So most of the EVs actually perfused into the host cells by either clarithrine or caveolin mediated pathway. So as I said, the endocytosis for an nanocyte vesicles is very much anticipated, but we next try to see whether these are kind of, because the lipid compositions of many of the lipids which is present in the bacterial membrane are fusogenic in nature. So we are interested to see, is there any chance that, like the virus, when they enter to the host cells by cytosolic, by directly fusion with the membranes, whether similar kind of attributes present in case of the when the OMV is getting uptaken by the cells. So yes, so we, and for that work, Pradeep has helped me a lot. Actually, it's done in his lab. So different assay, which are very standard assay to determine determine the fusions. So we use OMVs and then probed liposomes by freight assay, and then dye dequenching assay, and also simply content mixing using sulforodamine B. And then in all three different assays give sufficient evidence, enough evidence to claim that there has a that OMB has a potential to enter inside the cell via membrane fusion, which is really interesting, because as a bacteria they cannot utilize the fusion pathway. They only and mostly they enter inside the host cell by endocytosis, either it's a phagocytic cells, macrophage cells, or simple epithelial cells. But as an end product of the same bacteria, OMVs can facilitate the cytosolic entry. So that is, we feel that it's quite interesting. So with all those compositional aspect, their nanosized their size, their biophysical characters, and their functional characters, for example, fusiogenicity or intracellular uptake, led us to characterize the overall functions into two categories. One, whether this is an offensive striker for the microbes, because does microbes utilize those OMVs as a mode for further virulency or further pathogenicity, because of all those properties, all those functional attributes like nutrient acquisition, host tissue disruption, biofilm formations, delivery of the virulency factor. So they certainly contribute bacterial pathogenicity. But on the same time, since they're nano-sized vesicles and they can be uptaken by the host cells by the both pathways, endocytosis and cytosolic, can they be a good representation for host immunomodulations? So we can see that two facets of those. That's why I said bifunctional attributes of the EVs, which more or less we established, and previous studies also established these functions. But whether they can be utilized as a potential immunoadjunctive potential, we can see or not. So for that we yeah. So we also further we see in the context of campylobacteria whether they can facilitate bacterial pathogenicity. And we can see from this picture, this micrograph, it is actually with the whole bacteria, while the campylobacteria infect or co-incubate with the avian cells, which is chicken cells. They do not do any harm, while in case of the human intestinal 407, the membrane architecture completely gone, which said that bacteria can invade the host cells, and because of different compositions which are highly toxic. For example, this O specific chain for the LOS, lipo-oligosaccharide, which doesn't have that, but LOS is the integral component of their of the Campylobacter jejuni, which is considered as an endotoxic factors. And we also seen in case of the OMGs, these concentrations are actually a bit of higher range, that is 0.09 milligram of LOS in 1 milligram of total OMG protein. So which is sufficient enough to induce endotoxicity. Now, when we co-incubate with the OMGs with the bacteria, we observe the enhanced uptake or enhanced bacterial invasion of the host cells. So that clearly says that OMB can facilitate the bacterial virulency or bacterial pathogenicity, which come to a conclusion that make us to conclude that these OMBs are not the simple byproduct. They contain the functional signature for bacterial aggressions. They can disrupt the epithelial barrier and transfer antigen and virulency determinants into the local cells. And whether those local cells are immune cells or simple epithelial cells, that was our next aim to find. Now, if you see the major pathway, why we consider them as a very good immunoadjunctive agent or adjuvant effect or a vaccine candidate, is because they can enter inside the host cells, as I said, as we demonstrated that both cytosolic and endocytic pathway. So that is actually the requirement for an ideal vaccine candidate. They can stimulate both cytotoxic T cells and T helper cells, and this way they can cover the both arm of our immune system. So with this background, background studies and informations, we started doing an in vivo experiment where we used mice as a model, and those OMVs we introduced orally into the mice, and then those mice were challenged with the Campylobacteria. They received 3 immunizations, and then on day 28 infections, and then we sacrificed those animals to see the sickle load of the bacteria because they mostly reside in the cecum. And at that point, before the challenge, we also checked the antibody responses, so we can see a very strong local immune response elicited by those OMV immunizations in those mice, which is and all those secretory IgA which are raised in the locally in the intestine are found to be neutralizing. That means they're functionally active. Also, that affects the T cell responses, and we can see that OMV is encapsulated in chitosan, because we try to, I'm not trying to discuss about this approach, because we try to protect the OMVs from the host gut environment using a coating with a biopolymer which is called chitosan. That enhances that increases the CD3 T cell responses overall, and particularly CD4 and Th17 type of cells. We analyzed, and we found that they're significantly higher in compared to the other experimental groups, particularly the control group. So, so this is probably, I mean, a story can be ended at this point, but there are many questions with respect to the OMBs from the gram-negative bacteria needs to be addressed. For example, we cannot completely remove the LPS or LOS. That will not help forming the vesicular structure. So we are trying to see what could be the possible way to reduce the LOS or LPS content in the OMVs, because if we reduce to a therapeutic to a level which is approved level, then probably they can be a very good vaccine delivery strategy, or vaccine itself a vaccine candidate. So we are working on that. Now, the similar approach we applied for the Lactococcus lactis, and as I discussed, that EV's production from the gram-positive bacteria a bit difficult than the gram-negative bacteria. So the concentration is much more lower. We could not determine the actual numbers of how much EVs is produced, because that needs a specific equipment which is called nanoparticle tracking analyzer that can tell you how many vesicles are there. So without having that, we are also indirectly try to see what are the size and the protein concentration. So total protein concentration, actually we think that it represents the number of vesicles. So, but as I said, that for the gram-positive bacteria, a bit difficult. So how to resolve that issue means what could be the possible approach that we can utilize to increase the OMV biogenesis by the gram-positive bacteria. I'm sure not many literature is available in this aspect, because I don't know why people are not very much interested with the EVs from the gram-positive bacteria, possibly this low yield, or maybe other reasons. So what we tried to do is see that what are the different functions of the antibiotic, and what are the, because the composition of the EVs have the integral part of the bacteria. So whether antibiotic treatment of those bacteria in a particular target can help for the EV's biogenesis. So after reading this work is being carried out by Sushmita, and after searching a lot of literature, reading a lot of papers, what's it come to a point that that could be two possibilities. One is controlled inhibition of the protein synthesis, and the second is targeting the peptidoglycan layer, because this is the peptidoglycan layer is the main barrier for EV to come out from the inner membrane. So ampicillin is one of the beta-lactam group of antibiotic we have chosen, and chloramphenicol as a protein synthesis inhibitor, because they directly bind to the 50S ribosomes and prevents the formation of the peptide bond. When they bind to 50S ribosomes, they inhibit the protein synthesis. And the peptidoglycan of the bacterial cell wall contains some penicillin binding protein domain, which can generally targeted by the ampicillins. So that helps loosening this cross-linking. So whether that helps for the OMV biogenesis or not. So some initial study we have done, although it's inconclusive. When we tried the chloramphenicol, we don't see a very good pattern, means ups, downs, up, down. So there is no consistency, even for the total carbohydrate content. But when you use ampicillin, we can see some sort of indications that probably helps for more EV productions. And of course, the chloramphenicol and ampicillin dose what we use is much below the sub minimum inhibitory concentrations for those particular bacteria. So this is one of the things that we have been trying, how to increase the EV production from the gram-positive bacteria. And as in my earlier slides, I was talking about what could be the potential usage of the BEVs or bacterial vesicles in addition to and I was describing about different perspective of EVs use. So one of the perspectives that I told that to use as a bioengineering of those EVs or recombinant EVs. So for that, we utilized our own expertise in bioengineering bacteria, and we wanted to see whether the bioengineered bacteria, once they release the BEVs, they can carry the some of the target proteins, heterologous proteins. So the BEVs we isolated from those recombinant bacteria which we generated in our lab using two reporter proteins. One is mcherry, another is GFP, and you can see the bacteria once they are induced with nisin because they have the nisin inducible cassette. So whole bacteria turned either cherry or the green color. And we have also established protocols for how to express a heterologous protein on the surface of the bacteria. So utilizing all those three different bioengineered bacteria, we wanted to see whether the EVs released from those bacteria can carry the proteins, because those proteins are expressed in the cytoplasm. In this case, the protein is expressed on the surface. So it's a surface anchored versions of those bacteria. And we could see that recombinant, we could see the BVs which are released by the recombinant bacteria have signals, can show some signals of mcherry. For example, here you can see that mcherry EV has the emission is 610, and we can get a strong signal compared to PBS or non-recombinant EVs. And also by super resolution microscopy, we see the bacteria. We see the bacteria here, and there is bacteria as a Lactococcus lactis, and we contemplate that these are probably the bulged out based on the previous literature with the other bacteria. So we're still working on it to verify we can find the more similar type of budding out captured from the other fields or repeating those experiments. And these are confocal versus stain microscopy of the EVs, isolated EVs from the culture supernatant, and you can see the signals. But I think we need to work more on it. But at least what we can see from that, that bioengineering of EV, recombinant EVs can be made from a bioengineered bacteria. Okay, so and finally, what also we are trying to see whether any drug can be encapsulated inside the bacteria, inside those BEVs. So initially, we just started. We used doxorubicin as a drug, and we electroporate the doxorubicin into those EVs, and and we can see the doxorubicin signal from here. And this is the trit when we disrupt the vesicles with the triton, we can get more signal compared to the intact one. So that also give us some indication that it's also possible to encapsulate doxorubicins. So eventually we are planning to study whether dox-loaded EVs can be used as an anti-cancer modality in vitro, and in future probably in the in vivo models. So with this, the main conclusions what we studied so far with respect to the EVs are that they are heterogenic in nature. They carry different subcellular fractions of the bacteria. They can be internalized by multimodal pathways utilizing both endocytic and cytosolic pathways. They are the product of the bacteria that can facilitate bacterial pathogenicity, so they help bacteria for their own sake. On the other hand, host can utilize those as a canonical end product and good immunomodulatory agents. So if we use proper delivery strategy like oral delivery strategy, and there is a possibility that if we fine tune the bacteria with some sort of strategy, then we can induce the bacterial more BEV or bacterial EV biogenesis. They can carry the heterologous protein that we termed as the recombinant vesicles, and they can potentially carry many therapeutic molecules. So for further reference and the reading, I request all the students, those who are interested, you can visit my lab website. And these are the very relevant paper where you can get more information on how the OMBs are utilized here in terms of protocol and their functionality. And with this, I will conclude my talk today. And I want to acknowledge DBT and DST ACRB for funding our research, ISR Kolkata for infrastructural facilities, and my team, Dr. Ankita Singh. She actually started the OMB works with Campylobacteria. She is now postdoc in US, and current student Afroja and Sushmita. They are taking up these things. One from the gram-negative bacterial side, and Sushmita from the gram-positive bacteria, and of course my collaborator Pradeep for his continuous help and support for particularly working on this project, and he's also co-PI of the ongoing project that is that is for bacterial extracellular vesicles isolated from gram-positive bacteria. And Abhijit, of course, his student, he is now postdoc in US. So with this, I thank you. I want to thank you, all of you, again for spending your time in these odd hours. And now I'm open to take questions.
So, thank you, Professor Amirul. So, any questions?
Sir, I have a question. Hello, sir. I am Boygo, sir. Very fascinating talk, sir. Sir, my question is that the how are you loading the natural proteins within the vesicles and the artificial drugs or peptides? How are you loading them within the vesicles?
Can you repeat again how we are loading the drug or
Uh,
protein?
Both, sir. How are you loading it? Like naturally, how are you loading it, or are you loading it artificially?
So naturally loading means, let's say, make it clear. So there are two type of OMBs we talk about here. One is natural OMBs, which are extracted from the Campylobacteria. We didn't do any modification of that. This is just a natural entity. And the second part of my talk, where I talk about bioengineered bacteria, which releases the BEVs or bacterial vesicles, we term them recombinant vesicles, is, is the source is the bacteria, right? And those bacteria are are transformed with the plasmid, which carry the reporter proteins. And the plasmid what we use, it has an ability, it is a nisin inducible plasmid, so it can express the proteins there under the control of nisin inducible controlled expression system into the cytoplasms. So when, as we see that OMBs or EVs can carry the cytosolic fractions, so before even starting the work, we thought of expressing the protein directly in the cytosol will be much more, more sure for carrying the proteins into the BVs when they release it, rather than in the cell surface, because it's on the peptidoglycan, and peptidoglycan is not often come out along with the BVs. So it's not a good idea to use the surface anchored version of those recombinant bacteria. Rather, we chose to use the cytosolic version, cytosolic protein version of these plasmids. Is it clear? And for the drug loading, we just used, Afruja is here also, so we used electroporation method, simply the way we do for bacterial transformation using for plasmid. So it's electroporation with the doxorubicin drug and the BEVs.
Yes, sir. My question was like in the recombinant pathway that you told, like the proteins are expressed in the cytoplasm, right? So
Right.
how is it going from the cytoplasm to the outer membrane vesicles?
So so for the cytoplasm to the outer membrane vesicles, because when they bulge out, and at many points when there is a metabolic stress, all those proteins can accumulate on those particular area where they are tend to be bulged out. So so yeah, so if they accumulated more metabolic pressure, they help to push those proteins, or along with the other proteins, bacterial own protein, to the periplasmic space.
Okay, sir. So you are like inducing some metabolic pressure to
Yeah,
load these
we are
recombinant
not
proteins into the.
inducing metabolic pressure here. So we think that due to the expression of the protein, it's an additional metabolic pressure, metabolic stress for them.
Okay, sir. Got it.
Because even without metabolic stress, we can still see in the Campylobacteria OMBs, which are natural, we can still see the cytosolic proteins in the mass spectrum by protein, identification.
All right, sir.
Okay, so any other questions or queries? Okay, so I may have two. So
You have question?
Yeah.
Okay.
So no, it's not a question. Maybe queries that this when it is inducing the immune response.
Mhm.
So means you are means so you are giving the OMBs, right? So
Right.
these OMBs have some fragments of the protein.
Yes.
So that actually inducing the immune response. Is it whether I
Yes.
understand correctly?
Yeah. So is the protein also the different other other lipoproteins, other peptide, sorry, lipopolysaccharides, they are potentially, all are immunomodifier.
Hmm. So they are actually inducing the means immunogenicity. And
Immunogenicity.
how you are actually means how to quantify that means how much is their generating.
So if you inject those OMBs into animals, we collect the serum, means blood, as well as we also can collect the gut fluid, because most of the sick, because we are delivering it through the gut, it can be injectable, means there is no problem with that. So in both cases, if it is injected, then we are expecting to see good amount of immunoglobulin in the blood. Okay, so we collect the blood, we allow them to separate the serum, and then subject it to ELISA, and detect against the OMBs, or we can detect against the source bacterial proteins. Polylysate, for example, it's a Campylobacteria protein, so we coat the plate with Campylobacteria and try to detect how much Campylobacteria specific antibody OMB is generated in the blood.
Okay. Okay, got it. So another thing is that when suppose your OMB is bulge out, means
Mhm.
suppose it is gram negative for
Mhm.
simplicity. So from here it is just forming that and then
Right,
going out. So
pinch out.
now, for example, that for means when I'm just talking when it is bulging out.
Mhm.
So in when during endocytosis, it is known that this dynamine actually plays a key
Mhm.
role, means using the bulge out, means this
Mhm.
budding. So is it possible that these bacteria also have some kind of dynamine related protein, which may be probably helping this bulge out because you have the mass spec data. So
Mhm.
there you can maybe check that is it possible or some, because what dynamine does that it ties up and then during GTP hydrolysis, the means it goes out. Means this is after endocytosis. So similarly, is it possible? Just I was just thinking.
Yeah, I think it is possible to see whether those proteins are there in the population, protein population,
Hmm.
because there are a lot of protein. Most of the, if you see that, I think 60% proteins are unidentified protein,
Ah,
uncategorized protein. So there is a high chance that there will be possibilities is there.
oh no, but that may be also in the bacteria actually, because if it is cut, suppose dynamine, what it will do? It will cut it basically.
Mhm.
It will cut, so it may also reside in the bacteria. May
Yeah,
not go in the OMB. So
maybe.
bacteria has this kind of means that also can be checked that dynamine related protein is not exactly.
I got your point. Yeah,
Yeah, so that,
or similar protein.
or similar means related similar
Similar
means
protein.
that helps the budge out.
Mhm.
Okay, okay, okay. So if there is no more questions, so then let us thank Professor Amirul for his wonderful talk, and this means we learned many things. So thank you, Professor, for
Thank you.
your time, and also the iGEM team also they joined. Means this is their initiative, and since it's a virtual seminar webinar, so we could not offer you chai or coffee.
So we can
So
have it if you
if
are
you are interested, you can join in Bimolobu's shop
sure.
and have
Everyone, all are invited.
all of you if you want some. So you can have a, tea or coffee. Okay, then. Okay,
Okay.
then.
Thank you.
Thank you. Okay, bye.